There is increasing evidence that the dichotomy between vascular disease and neurodegenerative processes is an artificial one. We find, for instance, that if you take people with mild cognitive impairment, a clinical dementia rating scale score of 0.5, what was consistently seen in them was a breakdown of the blood-brain barrier in the hippocampal region. This might potentiate the deposition of amyloid and other neurodegenerative processes. We also find, when we look at genes, that there are genetic overlaps between what we traditionally considered vascular processes, such as small vessel disease in the brain, and what we traditionally considered classic Alzheimer neurodegenerative pathology with amyloid and tau in the brain. For instance, specific genes such as ATPH5, which is a mitochondrial-related gene, are shown to be associated with both small vessel stroke as well as Alzheimer's disease. We find that genes that impair function of the pericytes, like FOXF2, worsen the amount of amyloid burden in mouse models with the propensity to develop amyloid. And we find that in humans, it is not merely the coexistence of two pathologies that results in a clinical picture, but there seems to be a synergistic interaction between various pathologies. This is why one of the flagship projects of the NINDS in 2016 was something called MARC-VCID, whose goal is to develop biomarkers for vascular contributions to dementia and understand in individual people how much of a contribution comes from the different vascular pathologies, how much comes from the different neurodegenerative pathologies, and target trials better using a palette of biomarkers to specific individualized pathologies.